Good afternoon. My name is Bruce Wilson from Waters Corporation. I'm part of the Global Services Department, and I've been involved with supercritical chromatography since Waters started to get into the product line. I'm going to give you the inside story of how Waters developed our supercritical chromatography analytical product, UPC². This presentation will cover how we developed the UPC² from the beginning, including the internal creation of the product and commercial launch of the product, and how we sustain Analytical SFC, as well as opinions of future Analytical SFC directions. Before I cover this overview, I would really like to thank my other Symposium guests who are truly thought leaders in chromatography, supercritical chromatography, and supercritical extraction. My special thanks to Terry Berger, Mary Ellen McNally, J. David Pinkston, and Milton Lee. And if not for them, I would not be presenting here or training future SFC users and scientists in the art and science of SFC. Let's look at the 6 basic steps that Waters used in implementing Analytical SFC into the common laboratory. And the first is how we came out with the originals of UPC². And that was actually started out with Jim Waters and the Thar SFC purchase. Next, we talk about UPC² product development, how we went from prototype to product release. Next, we'll talk about UPC² technology innovations in SFC, and that's basically with the pump and the design of it with the cooling, as well as CO2 management, or what we call the convergence manager. During the development of our Analytical SFC product, it was very important that we also included the column chemistry for aChiral and chiral solutions. We'll be discussing a little bit of roadmap for that. Next, we'll talk about sustaining UPC² in Analytical SFC, and that's through training and support. This is true of any analytical product. You can start it, but without sustaining it, it will not flourish. Finally, I'm going to give you an overview of what I think is the current trends, which is really trending towards high-end mass spectrometry with Analytical SFC or UPC². Let's look at the first step, the origins of UPC², and the beginnings of the concept of Analytical SFC development for Waters. Waters' entry into SFC started around 2007 to 2009. And it was really larger pharmaceutical companies with existing SFC system wanting Waters to invest in an SFC company to provide a true one-vendor solution for analytical and prep SFC. Jim Waters actually introduced Waters Corporation and Thar Instruments together to make the introductions. In 2007, Waters and Thar Instruments actually collaborated to sell the ZQ-MS single quad detector with Thar SFC prep systems. This was Waters' first foray into analytical and preparative SFC. In 2009, Waters actually purchased Thar Instruments with the support of Warren Partz and Rohit Khanna. Warren Partz is actually Senior Director of Food Science and Environmental at Waters, and Rohit Khanna is a retired Senior Vice President of Marketing and Informatics. Waters decided to develop an Analytical SFC product during the timeline of 2010 to 2012. 2010, we actually decided to develop a commercial Analytical SFC system based upon UPLC technology. The key concept for Analytical SFC at Waters was to give customers a one-vendor total solution, which included a rugged, reliable, and robust equipment, integrated back pressure regulation, and built-in cooling for the pump, with no external chiller. Waters management wanted to remove customer purchase barriers based upon normal phase replacement, chiral separations and achiral separations, which was a product marketplace Waters had not been in before. Let's take a look at the progression of analytical SFC equipment from 2007 to current offerings. Back in 2007, a lot of the analytical SFC used custom modules that were proprietary and specialized software that was not combined with other analytical like LC and LCMS. In 2010, this was the prototype. We'll talk more about the prototype. It showed feasibility of developing a system. So this is where we'll talk about the design phase, alpha, beta, and pre-production. We'll also talk about the UPSFC innovation, or UPC², and how we manage automatic back pressure and back pressure functions, as well as the pump design for cooling and higher flow rates. Finally, this shows the progression of, to date, the UPC² with QDAMS, using a configuration with 8 column managers, a makeup pump, a pump splitter, and single quad MS detector. We'll talk more about MS innovations with SFC. Let's take a look at UPC product development and how we did it. So the pre-launch development lifecycle starts out with the design phase. The design phase is actually where engineering, hardware, software design, characterization, and electrical actually are started. The next step is the alpha phase. During the alpha phase, this is where product reliability, evaluation, as well as parallel column chemistry development of SFC columns are actually started. The next step from there is the beta phase. This is where applications become starting, scientific operations, as well as beta customers, qualification, service training, and serviceability. The next step is pre-production. This is where first customer ship are created, spare parts, preventative maintenance and qualification training, as well as base field installation procedures and general field installations. And the final is product commercialization, or the product launch. This is where sales training, marketing, our World Wide Web, as well as, of course, Pittsburgh conference in 2012. So this was the product life cycle that we did. One of the key areas of pre-development of any analytical product is the marketing requirements, or what is required to actually make that product. So first, we wanted to make sure that the analytical SFC solution we created, UPC², would fit with our software control platforms, which was Empower for LC solutions, including qualification abilities, and mass links for advanced mass spectrometry solutions. By doing this, it reduced the customer learning curve, and it's also scalable and networked in the Empower platform. Next, we're going to make sure we could use the same parts as much as possible as UPLC in the design and parts layout. By maintaining the form and fit, it gave same size factor. Familiarity reduces the barriers to implementation of a product, as well as we can reduce service time and service training because they already know what a UPLC parts are internally. And finally, especially in today's world, we can manage manufacturing challenges such as supply chain, which we all know that is a difficult item. So by having more volume of the same parts, we can reduce that time and cost for both internal and for our customers. Next, we wanted the column management and column chemistry options. So first, we wanted the ability to select multiple columns, anywhere from 2 columns, 4 columns, 6 columns, 8 columns, or 15. Also to give column heating and cooling, as well as a fit anywhere from 5 centimeters the whole way up to 30 centimeter column sizes. In addition, we wanted different chemistries, aChiral, and right now we have 8 scalable chemistries, as well as chiral, we have 3 chemistries that are non-scalable. So we wanted to make sure it gave chemistry options to support our customers, including let them use other vendors' columns. And finally, we wanted to give the customer detector options, including photodiode array, evaporative light scattering, mass spectrometry in the single quad, tandem quad, or quadruple time of flight functionality. Also to meet the wide variety of application detection needs for different customers. In the creation of an analytical SFC instrument, or again, any analytical instrument, you need to figure out what modules you need to develop brand new or modify existing modules. So with UPC², we actually did have to come up with a CO2 management module called the Convergence Manager, including income CO2 management, as well as a 2-stage back pressure regulation device. We'll explain that more in the next slide. We also had to redesign the binary pump. We had gas or liquid CO2 as a requirement, onboard pump head cooling, and we wanted co-solvent for solvent select valve. We just had to do a slight modification with the PDA detector to allow for the higher flow cell for higher back pressures, as well as modifications to reduce noise from CO2. In the column manager, we only had to really do tubing and valve changes to allow for the higher flow rates, as well as active reheating. And finally, in the sample manager, we could actually use the same injector. However, we did do some tubing ID changes as well as firmware for allow for the CO2 pressurization and depressurization steps. One of the true technology innovations of UPC² is the Convergence Manager, which is the CO2 management, which is more than a back pressure regulator. So, it has an integrated safety valve and vent valve. It also has the ability for a 2-stage back pressure regulation. So the 2-stage is actually a static cartridge, which is a small little check valve that builds the pressure to about 1,000 psi, then the automatic back pressure takes over. What this does is reduce maintenance and control. I'm showing you the static cartridge right now. This is something that is removable and can be done by the customer and does not require any technical knowledge. The next piece is it also has an auxiliary inject valve, which aids in depressurization of the CO2 during the inject cycle. In addition, there's a diagnostics ABPR health test. And finally, for CO2, there's a leak detection, which I'm showing you right now. That's that white disc. If there's any type of leak, it will shut off the system. In addition, there is a high pressure relief valve that if the incoming pressure from CO2 goes above 1200 psi, it will vent out and release, so it does not overpressurize the system. Finally, from a service perspective, all the components are pretty much replaceable from the front or on the side without having to destack the system. Only the boards, which are very reliable, would have to be opened up. This saves time and money. One of the major contributions to Analytical SFC is the UPC² pump design, which is called the CCBSM, and I'm showing you right here. So one of the major functions is that it's a binary pump, but it uses the same Acuity BSM design as UPLC and same chassis, but also same pump algorithm, which reduces the background noise because of pump pulsations. It's 6,000 psi, 4 mils a minute. Another innovation of the pump is you can do gas or liquid CO2. This is unusual as most preparative and analytical systems do require liquid CO2 only. The internal chillers, which I am showing you right now, are thermoelectric devices and eliminates the purchase of external chillers, which again saves time and money and size. We also added a PCB board for the chiller, as well as a 4-solvent select valve for co-solvent selection. When you create an analytical chromatography product, such as UPC² Analytical SFC, you want to do a lot of rugged reliability testing. So a few technology innovations that we had to do during our reliability testing, first was to test the ABPR and the Convergence Manager system. One of the major functions of reliability testing was testing the Convergence Manager, specifically the ABPR needle. The original ABPR needle was gold-plated and started degrading under high CO2 concentrations or high pressures. So to that effect, we modified the ABPR needle into a zirconium-based needle. This withstood high CO2 concentrations as well as higher pressures without any degradation effect. This was a very important step in the reliability of the Convergence Manager. Another area that we needed to check for true reliability was the CCBSM or the UPC² pump. The way we did that to reduce the amount of modules we needed for testing was we actually took A and B pump sides to test for CO2 and the chiller management. This way we were double up on the modules and reduce the amount of time for testing. Another area that's very important for chromatography systems and specifically for UPC² was the amount of solenoids and valves. So the way we did testing is we put multiple valves and solenoids into one test station, and instead of cycling every few minutes, we actually cycled every few seconds. We did this 24/7 to check reliability and the sustainability of the system over time. This is just some of the testing that goes on during the creation of an analytical LC or SFC instrument. As we evolved our UPC² Analytical SFC instrument, we also evolved our column chemistry. And we first came out with our first generation columns, which are aChiral chemistry offerings. Those came out between 2010 and 2012 that had 5 chemistry offerings, and they are also currently still used for installation and qualification services. There's analytical and prep. And the 5 chemistries gave you different selectivity. So as this chromatogram shows, the different selectivity gives you the different choices for method development. So again, it gave you different selectivities, which gave you different separations. As we evolved, though, we went to a second generation columns for aChiral, which are called the Torus brand. The Torus has 4 chemistry offerings, but a full method development path, and again offers analytical to prep SFC columns. So there are 4 different choices, and again a full method development path depending upon how your compounds are, whether they're basic, acidic, neutral, or mixtures. The Torus 2-pick is our go-to column for method development. When you create an analytical SFC system and chemistry, you want to give your customers a specialty template so they can develop their own method. So this is the generic template that we use using the Torus brand 2-pick. It's 1.2 mils a minute, 4 to 50% methanol in 3 minutes, and then 30 degrees C, 2000 psi back pressure. This will give a template to start, and then depending upon your column and your application, you can modify depending upon column selection. Then you change your co-solvent, your temperature, your additives, and your back pressure. Again, The idea of this is to give you a generic template to success. Another area that's extremely important for the sustainability of a product, and especially in the niche area as SFC, is applications, documentation, and user training materials. So one of the items that has been compiled from Waters is over 432 SFC and UPC references of a bibliography. These are ongoing publications that continually get updated as there's new documentation coming out. Also, there's direct and online UPC training seminars, as well as customers and service training manuals. Service training is just as important. So again, basic operation guides, including for Empower, translated into Chinese, as well as user and qualification manuals. On top of that, for training, one of the very important aspects is to get the new generation into SFC. So one of the items that we're using, and many companies are, as we all know, COVID is there, is UPC² remote training. In addition to that, virtual training is going on. On top of that, there's customer training. Service training is just as important to keep all service engineers up to speed on our products, especially in a niche area. Customer applications training, and also field chemist training. So these are all important parts of keeping an analytical product up to date and sustaining operations over time. The final area that I want to cover is what I believe is the future trends of analytical SFC. And definitely towards Waters, we're seeing a trend of analytical SFC and advanced MS detection, as shown here by one of our customer systems that has a UPC² with Xevo TQ-XS tandem quadrupole detector. Analytical SFC coupled with mass detection does require a splitter, and that splitter needs specifically designed to account for carbon dioxide and the desolvation of the carbon dioxide as it goes through the splitter. This is a technology that probably needs to be advanced more as we get into the higher, more sensitive mass detectors. Some of the science and technology that needs to be developed is how to account for splitters that do get blocked because of particles desolvating in the splitter and the decompression of the sample in the splitter tubing. I'd like to finish this presentation with some final thoughts about future trends of analytical SFC from my point of view. So first, in the education area, the only way to grow the SFC industry is really to educate our current and future analytical chemists in the science of SFC. The education and the use of SFC separation still requires hands-on training with the systems and not just virtual environment or using TeamViewer. SFC MS will continue to develop and advance as time goes on. In the analytical SFC customer application areas, we see continual focus and demand for cryoanalytical to prep scale-up for all geographies in the world. Food, cannabinoids, and traditional Chinese medicine are some of the analytical SFC-focused application areas. There's also an increase in demand from China and India CROs for purchasing analytical SFC. Finally, let's look at SFC versus Liquid chromatography from a future perspective. First, LC and mass spectrometry products truly outpace demand for analytical SFC products. In addition, the emphasis in bioanalysis reduces the focus on supercritical chromatography products. However, the fact that multiple chromatography manufacturers are making and selling SFC equipment validates the need for SFC systems. And finally, analytical and prep SFC instrument sales growth ultimately depends upon whether laboratory chemists continue to view SFC as a viable orthogonal tool to reverse phase. Thank you for taking the time to listen to this seminar, and I especially want to thank the James L. Water Symposium, the Pittsburgh Conference Management, my fellow symposium guests, Water's corporate management, customers purchasing SFC products because without them, we would not be here, and finally, for you taking the time to listen to this SFC and UPC² system overview. Thanks, and I'm open to questions.